Hello, everyone. I welcome you all in the sixth lecture of the course Cellular Microenvironment from Oxygen Diffusion to Gene Expression Analysis. In previous lecture, we already learned about what is gene, how gene expression happens, and what are the mechanistic way of understanding the gene expression, and what are the significances of the gene expression inside our cells. But we also learned about how translation and transcription happen inside the DNA, inside the nucleus. Uh, Not in the nucleus, but again, translation happens up, in the ribosome, which is there in the cytoplasm. So in today's lecture, we are going to understand the more deeper concept of, understanding the gene expression, like how does a cell turn gene on and off. And in doing these things, we will understand what is histone protein and the structure of histone protein. And then we will also understand the chromatin remodeling. Then we will also understand how the chemical modifications of histone can happen, and that facilitates the uh, easy opening of the chromatin for the gene expression analysis. Then we'll understand the DNA methylation, histone modification, as I've told you, transcription factor, and non-coding sequence of the DNA. So without wasting our time, let's try to understand the thing. So for understanding these things, first we need to understand the DNA. That is one of the fundamental unit of the cells that is responsible for uh, storing the genetic informations uh, for the cells. So when we talk about the cells, then there is a fundamental unit that is the DNA, and that is the incredible scale. I mean, we are going to understand the length scale of the DNA here. So when we see the DNA, so the length of the human genome is 2 meter. Or you can say the length of human DNA is 2 meter if I'm talking about one DNA. And the length of, I mean, the diameter of the nucleus is only 6 micron. So it looks very strange that how a 2 meter of length DNA can get accommodated in only 6 micron of the nucleus. So if you see that if the DNA inside one human cell is about 2 meter long, then how can it fit inside a nucleus? And that is the only about 5 to 10 micrometer in the diameter. And this is like trying to fit a 20-kilometer, long rope inside a tennis ball. Obviously, DNA cannot, remain stressed out, but it must be packed. And the cell solves this problem using histone protein by itself. So this is the, size of the nucleus that you can see in which the DNA will get exposed. And already we have seen that in the total number of base pair in the human genome is 3 billion. That is itself is a huge quantity. And when we talk about the molecular scale of DNA, it is very tiny, and you can see that the double helix diameter is approximately 2 nanometer. So what happens? It is just beyond the imagination. So if we stretch the DNA from all the cells in our body, as I have told you, there will be 3 billions of the nucleotides inside the DNA. So you could extend for over 37 billions of kilometers, right? So that much amount of the DNA are there in our body, but that is different part. Definitely, cells are having some regulatory mechanism that will try to solve this problem. And in this lecture, we are going to understand how cells solve this kind of problem to accommodate this DNA inside the nucleus. So in order to understand that, first we should understand the histone protein and that is the structural foundation of the chromatin. That is very important. So if you see the histone protein, it is there is a small positively this is a small positively charged protein, and over which DNA is wrapped due to the negative charge due to the presence of phosphate sugar backbone. So what happens? These positive and negative charge attract each other, and therefore DNA naturally wraps around the histone protein. So if you see that, let's say this pink, this ball is a histone protein having positively charged. We'll show you why this is positively charged also. And due to the negatively charged of DNA, due to the Coulombic interaction, what happens? These DNA molecules can, wrapped over the protein, and then it becomes very easy for the DNA to get accommodated inside the nucleus. So therefore, DNA naturally wraps around the histone protein due to the Coulombic interaction. And so here we can understand with a simple analogy that if a histone acts like a spool, you can see here, uh, then the DNA behaves like a thread, and this will wrap around this spool. But one histone is not enough, right? Because large number of the size of histone would be very small, and DNA's length is 6 meters, so it is 2 meters, so that is very long. So that requires eight histone protein that will assemble together, and this structure is called as the histone octamer. So now naturally you can see now the size of the histone will be large because eight histone proteins are getting assembled together and having different name like two number of units of H2A, two of H2B, two of H3 and two of H4. So total eight number of histone protein will assemble together and that is called as the histone octamer. Now the size is large, the DNA molecules will get wrapped over there and that will solve the problem for the accommodation of the DNA molecules inside the nucleus. And this is how this kind of analogy can be understood. Now, then there comes a nucleosome, two beads on a string kind of a structure. So as we have seen that now DNA molecules can wrapped over a histone octamer protein, and that structure is called as the nucleosome. And now the diameter of this nucleosome is approximately 10 nanometer of the diameter. So earlier now you can see the DNA molecules were 2 nanometer long. Now it is getting, the length scale of only 10 nanometer after the wrapping of the DNA over the histone protein. So now that solves the problem, and that is this structure is called as the nucleosome. So then we can define a nucleosome is the basic structural unit of the chromatin, and one nucleosome contains one histone octamer and the DNA wrapped on it that you can see over there on the screen. When many nucleosomes are connected, they look like a beads on a string kind of structure. So lots of DNAs are there, lots of histone proteins will be there in our cells, and then you can see that this structure looks like beads on a string kind of a structure when many nucleosomes are getting connected with each other. And this is the least condensed form of the chromatin having diameter is 10 nanometers. So this structure will be called as the chromatin, and that is the least condensed form, right? Now that this bits on a string kind of a structure finally will get condense, and then you can see that this will get folded, and this folded structure is called as the chromatin. And now you can see this large number of histone proteins are there and over which DNA molecules are wrapped, and finally it solves the problem of the accommodation of the DNA molecules inside the nucleus. So I hope this is clear to all of you how DNA molecules, such a large a structure of the DNA that is 2 meter, that can get accommodated only in the 6 micrometers of the nucleus, right? So this is how cell solves the problem of accommodation of the DNA molecules. Now coming to the next part, that is very interesting. That how I mean, what are the existence of the chromatin in our nucleus? So a chromatin can exist in two forms. So first is like if we can see in the first case, it is like open chromatin that is also called as the euchromatin, and closed chromatin. Second one is the closed chromatin that is called as the heterochromatin, and this is where the gene expression begins. And this is the fundamental concept where you will actually understand how gene expression can happen. So when we talk about open chromatin, you can see that chromatin is open. That is the least condensed form of the chromatin. So it is completely open. So now let's say if any gene expression happens here in this particular unit, what will happen? The RNA molecules, this any site, if we talk about open chromatin, any site which shows the gene expression, then RNA, it will be very easy for the RNA polymerase to access that particular site because this is open now. So there is no hindrance. So RNA polymerase will come, it will read the DNA strands, and finally it will, transcribe that information into the mRNA. Is that clear? But if you see that about the open chromatin, you can see that nucleosomes are, relatively far apart. DNA is exposed, then it will be easy for the RNA polymerase to get bind, and a transcription factor can bind very easily, and then it can transcribe gene into the mRNA. So if you understand these things, we can understand this with some simple analogy, like it is open book where easily any page is easily available. So let's say if you want to gather some information, so you can directly go to that page and you can get that information. And this is what happens in case of open chromatin, where it is open and obviously RNA molecules. I'm sorry, RNA polymerase enzyme wants some information, and that information's finding of that information should be easy if it is open. But when we talk about the closed chromatin, here you can see that the uh, nucleosomes are very tightly packed, right? So now there is no space for RNA polymerase to enter and access that the part of the DNA where the gene expression has happened. So in this, you will see that nucleosome, becomes tightly packed. DNA is buried, and then RNA polymerase cannot bind, and transcription factor cannot bind also, and finally gene expression or gene cannot be transcribed very easily. We'll study about the transcription factor in more detail in lecture 9, so don't worry about that thing. But this is how you have to understand the analogy. So if we see a closed book, then first you have to open that book, and then finally you can access any pages if you want to get some information. And that is what that happens in case of closed chromatin. So that's why in closed chromatin, gene expression will be very difficult, or transcription of gene into mRNA will be very difficult. So the important part of this discussion is that if you see here that if there is any gene expression happens in the DNA, then then the nucleosome has to open, or the chromatin has to open, right? And that is what we are going to understand. So because you can see here, due to the Coulombic interaction, the DNA molecules and histone protein are connected with each other. But if it is closed, then the interaction is too much. But if we want to open that, structure, then obviously the interaction, the Coulombic interaction that is happening between, or that is existing between the histone and the DNA molecules, you have to break. Either you neutralize the charge on the histone protein, or you another way is to neutralize the charge on DNA molecules. And there are some chemical modification that you can perform for the histone as well as DNA, and you can do these things, and you cannot do these things, but cells can do these things, right? Cells can perform all of these things to open the chromatin, and finally it can be transcribed into the mRNA. So now we are going to understand that thing. So as we have seen that why does chromatin become tight? Because if a cell wants to express a gene, then definitely it will open the chromatin and then gene will turn on. If a cell does not want to, or if a cell wants to silence a gene, then definitely it will close the chromatin, and then there will be that gene will be turned off. So this is how these things work. But how does a cell open a chromatin? That is very important. So in order to open the chromatin, definitely you need some enzyme, because all of these, function or work inside a cell that has been done by some enzymes. And that is why, first cells transcribe, first RNA transcribe the gene expression into the mRNA, and then finally, the translation happens from, RNA to, protein, and then that protein actually works for the cell. So similarly, here you will also see that here the work of the cells is to open the chromatin. So that obviously that will be performed by some enzyme. And here that enzyme basically what happens, that moves nucleosome far apart to each other or far from each other, and that slice nucleosome that can also remove nucleosome, and that also add nucleosome. And finally, after performing all of these, I won't say all of these steps, but again, that can perform this kind of a steps modification, or that can perform this kind of work, and finally, it is called as the chromatin remodeling. So let's say we can understand these things with this simple analogy. Let's say if there is a home, or if there is a room filled with furniture, and if you want to access this part of the room, then it will be very difficult because lots of furnitures are there, and then it will create some kind of hindrance. But let's say if you remove this furniture somehow from the middle, then now it becomes accessible. This area, let's say this window becomes accessible. But if you remove all the furnitures from the room, then now you are completely free to roam here and there, and this window is easily accessible. And this is how these things can be done by the any enzyme inside the cells. That is the respective enzyme. So first chromatin is closed, Now what will happen? This enzyme will come and it will try to slide the nucleosome, or it will try to remove the nucleosome, or if it is necessary, then it can add the nucleosome also. And finally, it will be able to, open the chromatin. Now, once the chromatin will get open, now it is easily accessible for, RNA polymerase to, read the part of the DNA where the gene expression has happened. And finally, once it will read, then it can transcribe that information into the mRNA. I hope this concept is clear to all of you, how cells open the chromatin, and this is very important concept. And I try to explain, in the best possible way while using this, room in furniture in room kind of analogy. Next, coming to the next part, that how histone modifications can happen. That is the most important part of this lecture, and also I can tell you this is the most important part of this lecture series of the cellular microenvironment, because if you are able to understand these things, the modification, then all the things will be very easy for you. So if we see the histone structure, then you can see that it is having a long tail, and this long tail is a kind of, amino acid, because protein is protein is made up of sequence of amino acid, right? And obviously this tail will be some amino acids. So if we see here, this amino acid can be lysine, arginine, serine, and threonine. That depends upon, what kind of amino acids are there. Then what happens? This amino acid is just a chemical structure. Definitely, there will be some chemistry behind that. So, you can modify this, amino acid using various chemical reactions like acetylation, methylation, phosphorylation, ubiquitination, something like that. Okay. And in order to do this kind of chemical modification, you can, neutralize the charge over the histone protein. And once you are able to neutralize the charge over the histone protein, or if the charge get neutralized, then definitely the interaction between the DNA and the histone protein will get decreased or reduced. And that will facilitate for the opening of the chromatin structure. And if let's say, for example, lysine is there, and in lysine you can see this is connected with the histone molecules, and there is a positively charged, right? Now, if due to this positively charged and DNA molecule is having negative charge, right? So what will happen? This will, facilitate the Coulombic interaction between the DNA molecules and histone protein. Now, if by any means, if you are able to, if cells are able to neutralize this positively charge of the lysine, then what will happen? The Coulombic interaction will get reduced. And due to this reduction in Coulombic interaction or Coulombic force, what will happen? The chain or the chromatin structure can get open. And this is how some, obviously, as I've told you, this positively charged neutralization will happen using histone acetyl transferase, enzyme, and that comes and it transfers an acetyl group. So this acetyl group is called as a CS3 double bond O minus, and this is a negatively charged group. And this, as I have told you, acetyl transferase enzyme will provide this kind of, functional group. And now this functional group will attack our NH3 molecules, and finally it will neutralize. And after acetylation, you can see this will form NHCOCH3. So now your histone protein got neutralized. Once it will get neutralized, then definitely it will be very easy. For earlier, what happens? There was a strong attraction between the DNA molecule and histone protein. After acetylation, what will happen? Due to neutralization of the histone protein, or obviously charge will get decreased on the histone protein. Now there will be a weak interaction. And due to this weak interaction, what will happen? Now the remodeling of chromatin will happen very easily, and finally it will be easy for the another enzyme to open the nucleosome, as I have told you earlier. And once nucleosome will get open, then RNA polymerase will come. It will, get binded with the specific region of the DNA where gene expression has happened. And now that informations can be transcribed into, mRNA. And finally, once mRNA transcription happens, in next lecture we'll see how that transcribed mRNA will get translated into the functional protein. Okay, so I hope I tried my best to let you understand how cells turn on a gene for the, transcription of, express gene into the mRNA. Okay, so I'll stop this lecture here. In next lecture, we will understand one of the another very important concept of this lecture is that how does DNA become a mRNA, right? Or you can say that how RNA transcript or how expressed in the DNA can be transcribed into mRNA. Okay, so we'll go in more detail how RNA polymerase comes to bind with the DNA, who is responsible for binding all of these things. We'll also understand how this reaction happens, that initiation, elongation, and termination, all of these things. How also we will also understand the you know the strands of the DNA. There are two strands in the DNA. We will call, there will be one coding strand, there will be template strand, all of these things. So next lecture will be also very interesting. So I hope you are getting some fundamental understanding of all these molecular biology techniques, and ultimately we'll converge these techniques to the instrument like RT-PCR, gel electrophoresis, and all of these things. Okay, thank you. I'll stop this lecture here. See you in the next.